So, Dan, can you tell me what are the advantages of the test to your patients and physicians?
So the calprotectin test offers a rapid, a relatively cheap, non-invasive method to diagnose and monitor patients with inflammatory bowel disease and reduce the need for colonoscopy, and that has benefits both for patients and for the wider health economy.
That's great, Dan. So can you explain how your physicians actually use the calprotectin results?
So the calprotectin test is used by primary care physicians as part of a diagnostic pathway to differentiate irritable bowel syndrome from inflammatory bowel disease. It's also used in secondary care by gastroenterologists to monitor patients with known inflammatory bowel disease.
Susanna, from your experience, what are the essential requirements for a calprotectin assay?
It's important for us that the assay is accurate and precise and shows good reproducibility. We have a lot of samples coming through, and therefore it's important that the extraction procedure is simple, and also that the assay can be used in an automated system and has IT connectivity. We feel that the BÜHLMANN assay provides these for us.
What you've said, Susanna, some of the biggest challenges for calprotectin testing is actually in the pre-analytical stage. With this in mind, can you explain how you quality control the extraction phase and the assay?
Yeah, we have created our own internal control to verify the extraction process. What we've done is created a pooled batch of previous stool samples, and we've run this every time we've run the assay, together with the internal controls for the kit. We've also used an external quality assurance program to compare our results with other laboratories throughout the UK.
How many samples do you get from that?
We have 3 samples sent to us each month.
How do you receive and store the samples?
We receive the samples at room temperature, and then they're stored in the fridge.
Can you explain how you validate the accuracy of the method and how does it perform compared to other methods?
So we validated the accuracy of the method by comparison to the previous method that we were using in the laboratory, and by comparison of results to a different laboratory. The method performs quite well on external quality assessment in our hands, and the BÜHLMANN assay does run with a slight positive bias compared to other assays that are used in the UK.
So how long have you been using the BÜHLMANN fCAL ELISA here at York?
We've been using the BÜHLMANN fCAL ELISA for about 10 years, so we jumped in pretty early.
So what's your experience with it? How long have you been using it?
My experience is that I've been using it for about 2 years on a day-to-day basis, extracting samples every day and then running tests 2 to 3 times a week. I also am responsible for maintaining the equipment, updating the standard operating procedure and training staff.
Why are you using this particular assay?
We found that the BÜHLMANN assay gave improved analytical quality compared to the previous method that we were using. The batch size was about right for the workload that we were undertaking at the time. The assay is robust enough to allow single cut analysis and can be automated on the DS2, so those are all important advantages for us.
Would you consider an automated solution like fCAL Turbo in the future?
Definitely. Our workload has increased so much over the last 10 years that I feel we need a more automated solution like fCAL Turbo in order to cope with that workload better. One of the advantages we feel of the BÜHLMANN method is that you can choose the option which is most suited to your workload. So labs with a lower workload may find the Quantum Blue or the ELISA more appropriate. But for us, the workload is such now that the fCAL Turbo would be our preferred option.
Do you have any advice that you'd offer to somebody who's about to start using the BÜHLMANN fCAL ELISA?
In the lab, it's important to have a really good standard operating procedure for daily use and training of lab staff. It's also really important to have a good extraction procedure that's reliable, reproducible and sustainable for the number of samples.
From the clinical perspective, the quality of the pathway that you use is key in ensuring that the test is used appropriately, particularly with regard to safety netting of patients who may have persistent symptoms but negative calprotectin results. We'd also recommend a phased introduction So making the test available for expert requesters who may be monitoring patients with known inflammatory bowel disease first, and then considering offering the test to non-expert requesters for diagnosis at a later date.